Here's some quick definitions. First, we have the neuron. The neuron is a nerve cell that conducts impulses. Next, we have the synapse. That's simply the gap between neurons. Then we have the presynaptic neuron. This is where signals transmit across the synapse, and they come from this neuron. Now we have the postsynaptic neuron. Now this carries the signal away from the synapse. Finally, we have action potential. That's just a transient depolarization of the membrane down the neuron. So now we're going to go through how neurotransmitters diffuse across the synapse and how they impact our presynaptic neuron and our postsynaptic neuron. We have the action potential move down our presynaptic neuron. Just like this. And after that, we have, in response to the action potential moving down the neuron, our calcium channels open, allowing more calcium in. So here we have our calcium channel. That's on the very end of our presynaptic neuron. And they move through like so. Next, we have our synaptic vesicles. These are just little plasma membrane-like material filled with a substance called neurotransmitters. I'm not going to go in depth about what a neurotransmitter is as of now, but we will get to that later in the video. So, our synaptic vesicles don't just sit in our presynaptic cell. As a result of the depolarization, the vesicle fuses with the membrane, and via excitosis, the neurotransmitters are released into the synapse, and that's this gap between the pre- and postsynaptic cell. Now, as a response to these neurotransmitters being released, the ion channels in the postsynaptic cell open. And then, when these neurotransmitters bind, it causes an action potential to transmit down the postsynaptic cell. However, these neurotransmitters don't just go all the way into the postsynaptic cell. Through the process of reuptake, the ion channels of the postsynaptic cell close. And when they close, the neurotransmitters are released back into the synapse, where they are taken up again by the presynaptic cell. This is just a representation of the channel closing. See how it's just all one cohesive thing and there is no opening anymore. Now, I would like to move on to talk about the in-depth process of each neurotransmitter. So let's get started. So special thanks to Pearson Biological Sciences 6th edition, specifically inspiration from figure 41.3. Remember, you don't need the light to do biology, you just need some glow-in-the-dark paint and some imagination. Thank you.